in the Department of Ecology and Evolutionary Biology, which is very similar to what a lot of you do in fisheries sciences. Um, and years ago, I used to read about adaptive management, and as I got into malaria, I was inspired by adaptive management for natural resources management, and wanted to develop adaptive malaria control. So I'm going to describe what we're doing now in two locations. One is with Uganda's National Malaria Control Division, and the other one with the Bioko Island Malaria Elimination Program. To get started, though, I want to make a very big distinction, at least it's an important one to me, between scientific research and malaria analytics. When I say malaria analytics, I'm going to define it as the systematic analysis of data for purposes of giving advice about malaria policy or supporting malaria decisions. And I'm going to be drawing contrasts with malaria research as I go throughout it. Oh, one other thing I should say, throughout the talk, you'll notice there's a QR code. If you point your camera at the QR code, you should be able to, it will take you to a web page where you can download either the paper that's on the page, or in this case, it's my web page here at the University of Washington. As I go through this, and because it's, you're a bunch of people who use mathematical models in various ways, I want to focus on the role of mathematical ecology and mathematical epidemiology and decision support. In particular, I want to ask a question: whether our devotion to simple mathematical models is it's pervasive in scientific research, but is it appropriate for malaria analytics? So we're going to be grappling with this question: what makes a model good, and good for what? And specifically on parsimony. So Albert Einstein characterized the question as this: he said a model should be as simple as possible, but no simpler. So it's a compelling idea, but we got no practical guidance about how to do that. And so for me, this structure is like, we don't know how complex a model needs to be to do a task. And so we will sometimes have a need to go too far. If you don't sometimes go too far, you'll never know what was the appropriate level of complexity. And how do you know you've gone too far? Well, sometimes it just becomes obvious that there's more things in the model than you need. But you have to do that. You have to push things to that far extreme and then come back to get what you want. So I'm going to discuss a lot this other notion that's sort of contrasted with that, called robust analytics. And I'll say a lot more about what I mean by robust analytics. But it's this idea that if you're going to give policy advice, you don't want to be wrong, or you want to worry about being wrong. So you want to look at alternatives, as many alternatives as you can. And so to make all of this work, we developed some software, and the software is up, and I'll give you some links to it later, and I'll describe how we developed this. But in particular, we want to be worried as we're doing all of this and trying to do malaria analytics and grappling with complexity about the uncertainty as well, right? So this robust analytics is a way of giving robust advice that's sort of honest about the uncertainty. So the goal is to characterize and quantify uncertainty and then propagate it through the mathematical models. Right. So I just want to say that malaria involves a lot of questions involving sort of medical issues in human populations and human malaria epidemiology, as well as mosquito ecology and transmission. And I'm going to stay focused largely on the mosquito side because I think it's going to be more interesting to y'all, and because you are in a school of aquatic sciences, and this is a freshwater aquatic species. So here is the QR code for the talk if you want to download it, and I will show this again at the end. So on this topic of malaria being a sort of a human disease, it's important. This is there's a metaphor for malaria that was written by the late Joel Bremen, who was a friend of mine, called the ears of the hippo. So when you see the ears of the hippo, it's obviously an indication that there's a much larger beast under the water. And his point is that malaria as a disease is often underappreciated because it's so pervasive and it's a chronic disease. So the spectrum of malaria as a disease includes severe malaria, which will present sometimes with high fever, coma, respiratory distress, often leading to death. Or it can also present with moderate symptoms like fever or sort of mild disease. It's particularly pernicious in women who are pregnant for the first time. They get a very severe case of malaria associated with birth defects, low birth weights. It's a disease that has written itself into our genes. So there was an association noticed by JBS Haldane who said, look, there's probably, it's called the malaria hypothesis. Malaria looks like it's the cause of sickle cell disease. Now we have sort of very interesting studies that relate resistance to malaria as a disease to the presence of at least one copy of the gene in the haploid state. And so we have this very large spectrum of disease. And just one sort of note to tell you how big of a problem it is. In Uganda, where we work, about 30% of outpatients who come to an clinic and don't get admitted, about 30% of them are diagnosed with malaria. So that's, giving you an idea. This is a pretty severe problem, big problem. But I want to talk about it in more as a sort of a management problem. So human malaria can be understood as a set of loosely spatially coupled, managed, complex adaptive systems with exogenous forcing as weather and malaria control. And, when we talk about adaptive, we're talking about the evolution of resistance to drugs and to pesticides, talking about socioeconomic development, and we're talking about a range of other conditions that change, including climate. So we're talking about a very complex system, and the two agents that we're going to focus on the one on the left, this is a diagram of the mosquito feeding cycle and life cycle. So once an adult has emerged, they mate, and then they go through this cycle of blood feeding and laying eggs, with sugar feeding intermittently or depending on the species and the context, sugar feeding at some frequency with all of that. The eggs are laid in the water. They hatch into larval instars. This is a homometabolous insect. Sorry, holometabolous insect. And so that means it has 4 larval stages. It pupates, and the adults emerge at some point in the future. So it gives you the ability to sort of do interesting studies of mosquito populations in the aquatic habitats if you want to learn something about development over and the sort of populations of time. On the other hand, you have a parasite with a complex life cycle. So within the mosquito, in a blood meal, parasites are taken up and they're transmitted later in the bite, in saliva. In between, they have to go into the gut, penetrate the gut, form an ookinete, and then an oocyst. The oocyst, it sort of just leads to replication in the number of mosquitoes, and they emerge as sporozoites. There's a flagellated form that swim into the mosquito salivary glands where they're ready to be bitten. Now in the human, that bite inserts in the saliva these sporozoites that then migrate into the liver. Sorry, I'm a malariologist. This is obligatory. I have to tell you the life cycle. You have to know it. And then once they come out of the liver, they start replicating in blood, and some fraction of them, and that replication cycle is sort of, we call it the asexual cycle. It just goes over and over again, and that's where you develop very high densities of parasites that are responsible for most of the disease. And at some stage, it's sort of some of the parasites in that asexual cycle make a commitment to become either male or female micro or macro gametocytes, and they get taken up in blood, mate in the mosquito, and it all goes around and around. So the events that we're going to focus on are these blood feeding events, where and blood feeding behavior, where it's the blood meal that infects the mosquito and the bite that infects humans. Right? The other thing about malaria that's really kind of important is it's transmitted by Anopheline mosquitoes, but there are lots of species of Anopheline mosquitoes. One of the crazy early century malaria epidemiologists who went out said that he compared malaria to a game of chess, where it's like simple pieces, but they combine in these unique ways. And this Hackett's hypothesis about malaria being a sort of locally peculiar thing is one of the things that concerns us about malaria control, because it means that we're probably, since we're, if we're malaria managers, we're not interested in the kind of normative advice that they would give at WHO or kind of stuff that appears in the scientific paper. We need to know how malaria works here in this particular site. And this is one of the reasons why when we do analytics, we want to care about locally relevant details, which are sometimes very hard to discover, but they're probably what's going to tell us what's the right thing to do if we're making some of our decisions. So we're going to rely heavily on malaria theory, particularly theory that tells us how to measure malaria transmission, and how to understand malaria transmission dynamics and control. So just a note here, side note, this foot has caused me a lot of grief. So if any of you want to sort of make a complaint about the foot later, what we're trying to do is illustrate the processes that drive malaria from sort of very local processes, and you know, often Anopheline mosquitoes in particular like to blood feed on your ankles. And so this is why we put a foot up there. And everybody said it was a Monty Python foot. So yes, it's a Monty Python foot, and you can complain about that later. But the point was that we wanted to be able to scale up from these very local processes and understand malaria going all the way to sort of local or to very large scale processes. Now, for all of this, we rely on malaria models that started a long time ago with Ronald Ross. So Ronald Ross was the physician who was working in Hyderabad, India, and looked under a microscope, and he found mosquitoes who were infected and who had sort of recognizably stained parasites in the gut of a mosquito, confirming that mosquitoes were the agent that transmitted the malaria, right? And so he won a Nobel Prize for this, and within a very short time after this, Ross and Battista Grassi worked out the life cycle of malaria. It took some time before this idea that malaria was actually mosquito transmitted was widely accepted, but you know, a lot of people accepted it immediately, and then there was debate among other people. In the next 12 years of his career, though, 13, 14 years, Ross set out to develop a theory for the prevention of malaria. And he was, you know, because he had a Nobel Prize and he was in the military, and you know, he'd been in the British Empire, he spent a lot of his time sort of promoting the prevention of malaria, and he was also a polymath. So at the same time he was doing this, he was trying to develop mathematics to understand malaria transmission so that he could do a better job of giving guidance to places like Mauritius and Egypt about malaria control. So in 1908, in a book called The Prevention of Malaria in Mauritius, he publishes the first mathematical model of malaria transmission. And then in 1911, in The Prevention of Malaria, he publishes a model, and again, this is the version that appears in a Nature paper in 1911. So we have this sort of first mathematical model describing malaria transmission, and Ross admitted fully, I mean, this is the last paragraph of the paper, he says, we need to do more on this. This is just the beginning, right? And 50 years later, we have now investigations that have followed up on many of these concepts. And this synthesis that was performed by George MacDonald in the 1950s ties all of this stuff together and assigns names to parameters. And this is where this basic concept of R naught. So R naught actually comes from Alfred Lotka, who was a human demographer. Little known among many people, though, he also published a solution to Ross's 1911 paper in 1912 in Nature. And he had this long-standing interest in malaria. In 1923, published a 5-part analysis of MacDonald's model. So he's the one who invented R naught to analyze life tables for human demography. So it's very clear that MacDonald got his idea about where R naught comes from Lotka, probably from reading his papers. And it turns out that MacDonald was actually not very good at math. He didn't do any of his own math. It appears in other publications. What he did, though, is a very good job of synthesizing the math, assigning names to parameters, inventing new concepts, and applying them to malaria control. And this is what we now call a Ross-MacDonald theory, a very simple theory for malaria. Now, after Ross stopped working essentially on the prevention of malaria, he decided the next phase of his career, he was going to work on something that he called a priori pathometry, a name that clearly hasn't caught on. Nowadays, we would call it mathematical epidemiology. But in the beginning to one of those papers, he advocates this as a process. So he makes a distinction between statistical analysis and mechanistic analysis, which he called a priori, and a posteriori. And essentially what he's doing is laying out a roadmap for us to use mathematical models in combining with statistics to understand complex things like malaria epidemics. Okay, he was actually going for a full-on theory of all epidemics. And it's an interesting story as to why it didn't catch on, but I'm not going to go down that path today. And so after Ross and MacDonald, there was a proliferation of theory, particularly starting in 1970s. when malaria has moved away from, well, it was sort of global malaria eradication program was over, malaria epidemiology moved into departments of ecology. And then there was this proliferation of models where now mosquito ecologists start to get involved and make better models of mosquito ecology. Now we have tools to do sort of irradiated sporozoites. So now we care about, the spread of irradiated mosquitoes. And a lot of these things come in. So in 2013, 12 years ago now, it seems like a long time, we did a review, and by that time there were about 540 papers that we could identify that had used some form of mathematical model. And this is a schematic representation of kinds of things that people had dealt with in those models. One of the things about Ross and McDonald is that in McDonald and Ross's models, there's a single parameter describing mosquitoes. All right. And so I've shown this before, but one of the things that we need to do if we want to start to give advice, particularly about vector control, is we have to open up that parameter and make it something we can investigate more thoroughly and in greater detail. So this is going to be the theme for a lot of the talk. And let me sort of spend just a moment to talk about this one concept that came out of McDonald's work. It was a guy named Charles Garrett Jones in the 1960s, was very interested in turning the entomological parameters that appear in that formula for R naught. He wanted to turn them into a basis for a theory of vector control. And so I worked on this quite a bit, and it turns out that this formula for vectorial capacity is really complex. These are the parameters at the left, and it turns out to have three terms. It's the lifetime number of expected human bites per mosquito, so counting from emergence to death on average. Take that quantity squared. Why? Because it takes 2 bites to transmit, one to infect the mosquito and one to infect the human. Then we count from the moment of emergence. So lambda is the emergence rate scaled to the number of humans. And then we have to survive through this long incubation period where the mosquitoes are sitting in the oocyst replicating for 10 to 12 days before they come into the salivary glands. So that's vectorial capacity in a nutshell. And we wrote about this more than 20 years ago now. What we wanted to do, though, is to start to turn this in. So, let me go back. One of the things, though, is you can start to sort of get a basis for what mosquitoes are going to, how transmission is going to respond to control just by counting the number of times that some of these parameters appear in these formulas. And here you can see that G appears a lot because it's, in S once, in P once, so it's 3 times. So actually, there's a hidden one in lambda. The blood feeding rate F appears twice, and probably there's another hidden one in lambda because mosquitoes turn blood into eggs and lay eggs, so there's sort of some feedback process there going on. So anyway, that idea of sensitivity to parameters was really influential, but we wanted to ask a more directed question. So if I go out and I only manage to spray 50% of the houses, or I only give out 50% of the people a net, what's the relationship between coverage and the effect size? So what this forced us to do is to sort of break up the feeding cycle now into parts. So how long does it take to make take a blood meal? So what fraction of the duration of the feeding cycle, this is something that's been studied a lot, was actually due to blood feeding as opposed to resting and egg laying. So we break that up and we look at how blood feeding is modified by the presence of a net with this, truly horrible diagram, but this is what passes for great diagrams in malaria epidemiology. You'll just have to get used to it. And so now what we can do is we can write that formula for vectorial capacity again, and we can say, well, what's the effect size on potential transmission? Because that's what vectorial capacity is giving us. I'll come back to that in just a minute. And we take the ratio of those parameters before and after in that and get a sort of an estimated reduction or an n-fold reduction. So that was what we're trying to do to get so we can move towards giving advice to say to a program, if you can only give so many nets out, how should you do it? We also know that when you write out the formula for vectorial capacity, that it's linearly proportional. There's a linear relationship between vectorial capacity and something we do measure in the field called the entomological inoculation rate. So if you go out, you can try to measure the number of infective bites that a person receives in a day. It's what we think of as EIR. The methods for doing it, maybe they're not as accurate as we'd like them to be, but that's what we try to measure. And the idea here is that vectorial capacity is actually measuring potential bites or bites emerging from or bites, potential for emerging, while EIR is measuring bites received. So in a closed population, ignoring mosquito migration and the delays, there should be a pretty close relationship between vectorial capacity and EIR. And this gives us a basis for now taking metrics and estimating R naught. And now we have a basis for setting thresholds. So this was in 2007. And now we can take our measures of EIR and PR. PR is the prevalence of malaria, and we can start generating maps. Okay. And so this is something we published in 2011, where we've now the first global map of R naught for malaria that had ever been published. And we of course actually propagated uncertainty through this. You don't see it in this figure, but in the appendices, we do a great job. And we try to give you some sense in these down below. It's where we're not so sure. There are various ways of representing uncertainty, but now we have a sort of a basis for setting thresholds. So we've been doing this, and then we start looking at data at a very, very local level. And this is, so I want to talk through this study in a bit because it's very interesting. So this is a study on coastal Kenya. So you can see from the maps, or sort of in the east coast of Kenya, east coast of Africa, Kenya, and we're going down to a place called Kilifi, where Medical Research Center has a research facility, and they're doing intensive surveillance, medical surveillance of the local population. So they know every person, they've enumerated every person in every household. And now they're looking and they're saying, when these people come to the hospital, if they're feverish, if they have a fever, what fraction of the time do they also have malaria? So we call this malaria positive fraction. What you're seeing up here, and what I really want you to take away from it is if you look at the dots, some are blue, which means that people almost never come in with malaria from that household. And in red, they almost always come in with malaria. And what you're seeing here is, particularly in the upper left, you see a lot of blue dots really, really close to a cluster of red dots. And as you look down along the river, there's where you would expect to see, maybe there's a lot of water, you'd expect to see a lot of mosquitoes. You see clusters of blue sort of mixed in with clusters of red. So clearly our picture of malaria transmission that we've drawn at a global scale is maybe useful at some level, but it's missing some of the details that are happening at these local levels. And this is why we start to want to unpack some of these models and say, how do we do a better job of understanding transmission so we can give you a better idea of how to set thresholds or to set rational expectations about what's going to happen if you go to try to intervene with vector control.
Each of those dots, there's one
house. And it represents 10 years of data from a single house, because this is what you get when you do a demographic surveillance system. You get just enormous amounts of data. And by the way, this is the lead on this paper was Philip Bijon, and we worked on this together. It was a really fun paper, but the point is we were trying to say, well, this looks fractal. Okay, it's baloney. Fractals require many more scales of observation, but the idea, the metaphor was good. So anyway. All right, I'm going to now describe how we use theory to develop adaptive malaria control, which I'm going to crudely describe as iterative, robust analytics for malaria policy. So let me explain that in a bit. So malaria control, the kinds of questions that we want to answer are usually about tailoring interventions to context, monitoring and evaluation to find how things work. And this is always happening under a budget constraint. So if you say, well, ideally, what's the best way to distribute IRS or ITNs? The answer is, well, give it spray everyone if you can. But that's, in reality, there's not enough money to do that in your program. And this is what we keep encountering with the violent malaria elimination program. So there's a choice. You can do bed nets to some extent, but if you spend your money on nets, you can't do any indoor residual spraying, which is where we spray pesticides on the interior walls of houses. And in the African species, they rest on those walls, absorb the pesticide through their feet, and die. So indoor residual spraying has been the driver of most of the policies that have been used. And we can also do larval source management. WHO and Global Fund really don't like that, but programs really, really want to try it because they've tried to do these other things and they're still stuck with problems. And attractive toxic sugar baits. And on the other hand, you have interventions that you can do through your health system. So vaccination is now a possibility for the first time, really. And that comes with an enormous cost, and who gets vaccinated and how often do you have to renew it. And so it turns out that the vaccines are probably one of the most cost-effective interventions compared to other vaccines. but probably one of the least cost-effective interventions for malaria compared to other forms of vector control, which is just to say that we have an enormously underfunded problem, and we should be able to do more. But anyway, these budget constraints are affecting trade-offs not just among vector control options, but among different kinds of control, medical versus vector interventions. So robust analytics is, malaria analytics, I've said this before, let me now define it, is a systematic analysis of data for decision support or to develop malaria policy advice, right? So the end point of analytics is to tell you what to do, not to make an assessment of what we know. Okay, so that's the latter of scientific research. Malaria analytics is what to do. And now, robust analytics for malaria policy, if I'm going to do that, and I have to give guidance about what to do, most of what we learned about scientific inference was designed for research. So we need a bespoke inferential system that's bespoke in two ways. One, it's for malaria, so we're going to build it around malaria metrics and malaria concepts. And two, we're going to build it for decision support, right? So if you're going to be given decision support, how do you do that in a rigorous way? And that's what we mean by bespoke inferential system. I got to put my glasses back on just a second. Oh, okay. What do I mean by the advice being robust? I mean, I don't mean that it's giving you a definite answer, but I mean that the advice wouldn't change if somebody says, well, did you look at this? Because you've already presumably identified all the sources of uncertainty, all the other reasonable ways of doing an analysis, and propagated them through your analysis. That's the idea of robustness. It's not that the advice gives you a certain answer, it's that I can tell you, I'm going to give you an advice about what to do, but I'm also going to tell you what other alternatives were and how well they were supported. And it sets up that next step where we say, well, if I give you a bit of advice and I'm uncertain about what to do, that advice is going to come with a bit of advice about what to measure, so that the next time we come around to do this again, we can reduce our uncertainty in that policy route. So a big emphasis here, and I've said this before, but I'm going to say it again, is to characterize and quantify uncertainty, and then to propagate that uncertainty through the analysis all the way to the endpoint of the malaria policy advice, which means you're going to probably be doing a lot of computation. You're going to have to be doing things in pipelines where the analytics get replicated thousands of times. And each one of them is going to be, using a lot of computer space. All right, so within that, then adaptive malaria control really makes sense as an iterative, robust analytics with this little loop where you think about what to measure, so the next time you come around, you can do a better job of it. So the core idea, we had to sort of, I'll be really honest, I got into this, I had this gut sense of where we needed to be, but we had to sort of develop a methodology that could be replicated in different places. So we had to fill in the gaps and give names to things that, or borrow other people's names for things and give them new definitions so that we could describe what it was we were doing accurately. And one of those terms that's become indispensable is this idea of malaria intelligence. What do I mean by intelligence? So if information is data that's been giving meaning through some kind of statistical or other analysis, then I mean intelligence is information that we need to make good decisions. Right? So I'm going to go looking for malaria intelligence. I'm going to go try to develop these very stable assessments because it's the same quantities that we're going to need over and over again, and we need to learn about these in situ over time. Right? So if we're going to build malaria intelligence, it needs to be timely. We need to start from the endpoint of the questions and work back to say what's the data we need to fill into this. We're going to identify these, I'm going to call them checkpoints, but they're informative metrics, things that we could design a study and measure them so we can check how accurate our assessments are. We don't want to just be sort of always in the dark. We want to do that. And then the other thing about malaria intelligence is if I'm going to be making policy for a country like Uganda, I can't leave any place out. So if I don't have any data, I have to find a really good way of if I don't have any good high quality data, I have to use low quality data, which I'm always going to get through their health management information systems. And I'm going to have to develop algorithms that they're honest about the uncertainty because I have to make a recommendation for everywhere. So that's one of the nasty things about, actually doing policy is you're bound to make a recommendation. And if you say, well, I'm just not going to do that, well, you're still actually going to make a policy recommendation that's going to affect something. You just weren't really sure what you were assuming about it. Right. So you can't really get around the fact that if you're going to do policy at a national level where you're weighing trade-offs and tailoring interventions to context, that you have to be making some assumption about every place. So you may as well sort of be very rigorous about it. So that geographical completeness is a kind of a very important thing. So this malaria analytics led us down this other pathway. So what does it mean to be a professional malaria analyst? And I was very shocked to discover that nobody has really ever thought of the question, or at least I haven't seen very good discussions of what it means to be the kind of person who sits in a malaria office. What do you do? What are your activities? All right, so we had to do some of that. So I just want to make this point, and it's partly to motivate malaria intelligence, but it's also to discuss the kinds of things that go on in malaria offices. There are 3 temporal epochs that we want to think about. And if you keep this clear in your head, it makes it easier to decide what you're doing. So there's the past where we get all our data. All of our statistical analysis is about the past. There was one past, never going to happen again. So I can't go back and do an experiment in the past, right? But this is where I'm going to make assessments where statistics get used. I do model fitting. I'm going to do history. I'm going to sort of use it to do monitoring and evaluation. So how well did things work in the past? The next, though, is this counterfactual past. It's a very important thing that we use in policy where we have to say, what would have happened if I had done something else? This counterfactual past ends up becoming really incredibly important in advocacy. How many lives did you save, right? As opposed to how many people are dying of malaria right now. And this becomes important because if you go to someplace, so we wrote about this in a paper called The Sisyphean Problem of Malaria in Zanzibar, where after a lot of effort, malaria had come down and people were complaining about all the money they were spending on malaria in Zanzibar. Our point was, yeah, well, take it away and see what happens. You're not paying for the cases you see, you're paying for the cases you prevent. And it's a very important concept to reiterate over and over in these policy debates where people are weighing malaria, spending on malaria that's happening now against TB that's happening now. You see a lot more TB. So you have to continually make this case that if you're doing something, you're saving lives. The present is where all of our decision gets made. But our data take us up to a few weeks ago or a few months ago, and we need to actually do this nowcasting where we fill in the gaps. And we ask questions about management questions like, are we in outbreak? So outbreak analysis is always about the present. And if there's a crisis happening, we need to be able to respond to it. This is also where we sort of make continuous statements about where we are, because if we're not doing that, we can't actually say where we need to go. And then, of course, all of our decisions are about the future. There are many possible futures, no data to describe it. So we have to find something to do to make decisions about the future, and that's involves forecasting and analysis of the past. So what we end up doing is we end up building these bespoke decision support systems in every place. So it's a methodology. It's not sort of we go in and we look at the data, we develop these malaria intelligence systems, and some of the components are shareable across sites and some are not. Now, to make this work right, to do all of the things I've said, we need to be nimble, right? So you sitting in a policy office, from 1 minute to the next, the discussion could change. And if you can't come back and provide policy advice in a reasonable amount of time, you just won't be useful. So you need to be able to change the kind of thing you're doing. And this is one of the reasons why we didn't want to go down this route of using individual-based models, because they take a long time to build and calibrate and turn advice back. It's usually in the order of months, and it's expensive in terms of computation cycles. So we wanted to be able to use simple models first, and then later do complicated models with the analysis. So we wanted to be able to scale complexity. And we also, in doing this, we wanted to sort of address this question. My advisor, Simon Levin, has this very lovely essay about intertidal ecosystems, where he talks about relevant detail. How do so many species coexist in the intertidal systems? And we can extend that idea of relevant detail to these. What details matter? So I'm going to go through this very quickly. I apologize. I'm also running out of time faster than I thought I was. So what we did is we built a mathematical framework so that we could take any models and do plug and play. We turned it into software, which is up here, Simba. What does the Simba do? It actually sort of cuts malaria into three big pieces. Okay. So there's aquatic ecology, what the mosquitoes, the immature mosquitoes are doing in the aquatic habitats. They're adult mosquitoes. They have ecology and infection dynamics. And those are connected to the ecology through an egg laying algorithm. So eggs go in, adults come out. And then there's an adult human ecology demography and infection dynamics. And those are connected through this blood feeding algorithm. And then we allow modification through all of these through this thing. Now the software works, it's coherent, and we think we've rooted out all the possible errors. It's available now for download through GitHub, and you can actually use it. And it's a fairly interface to start building models. It comes with a rubric for model building where we identify the questions. So, on the left, you sort of build a model. And the first thing you mean is you're going to sort of define a state space and define flows among states. So we take those as our sort of atomic units. There are many possible ones, modular, so we can pick one. Then we need to parameterize it. Then we need to ask questions. How many habitats are they? Where are they? We're going to carve up space into patches for purposes of understanding adult mosquito behavior. Then we're going to make a rule that says wherever humans live, they get to spend their time, right? So then we have a basis for doing spatial dynamics and so on. I could actually spend the whole time talking about this. I won't. But we now have a basis in software so that you can actually step by step go from very simple models to very complicated models, very realistic models, one step at a time. And this makes it possible to build up very large suites of models. So for the rest of this talk, I'm going to talk about adaptive vector control. I don't know how much time I have, not much. Where this is an island. So I'm looking now at the west coast of Africa. We're talking about Bioko Island, which is this blue dot that you see near Cameroon. And it's an island with volcanoes. And if you look on this, you can actually see that the middle part of the top end of the island is one big volcano, and a lot of people live around it. Almost all of them live at the north end of the island. This is an OpenStreetMap view of the north end of the island. So if you drive around, what does it look like? And the south end of the island is another volcano with a national park and some tourist attractions, some beaches. You can go swim with turtles, some nice things there. It's a very corrupt country in terms of sort of indices of corruption. It's an oil. Most of the economy is actually driven by oil. Before that, it was just one of the poorest countries in the world. So it's a very interesting place to work and interesting as a usual. So this is where 85 to 90% of the population lives at the north end of the island, and the program has mapped out every house. This is the map of population density at a 100 by 100 meter resolution. So we have very good data. And of course, you can see on this where the humans are. And now we have a larval source management program that we've implemented. And so we can tell you where we've found aquatic habitats. We don't know where all of them are. Of course, it's very difficult to know if you found them all, right? And now we have these very serious questions that we're starting to get at. Oh, let me make one more point. The total length across this part of the area with about 300,000 people is only about 18 kilometers. And if you're looking at this downtown area, it's 5 or 6 kilometers across. And that's where almost everyone lives, in that downtown area. So we're talking about a malaria control problem in essentially an urban area where people can walk around and get from one end of this big city to the other pretty easily. And so there's a lot of human movement. And we need to know where the mosquitoes are moving around, right? So I'm going to talk to you about a couple of things that we've done over the years and how we've done adaptive vector control in all of this. So the first question is about the transmission dynamics. And I told you that, so we don't want to assume things are well mixed. We want it to be spatial, but we also need to understand how mosquitoes are moving around. So how far do mosquitoes move? Why do they move? What spatial scales are appropriate for modeling malaria transmission for purposes of evaluating vector control? What, if anything, is a malaria population? do we have multiple populations on the island, or is there just one big well-mixed thing? What, if anything, is a focus? Is there focal transmission, right? And we're coming to believe that there actually might be a lot of foci on the side. And then, is all malaria transmission focal, or is a place like this some combination of sort of weirdly, you know, poorly or well mixed people moving around with sort of little, maybe hot pockets? I'm actually looking for corporate sponsorship, so we use the hot pocket term. Sorry. And then we're also interested in sort of combining vector control. So if we start doing things like combining indoor residual spraying and larval source management ITNs, which is actually what we're doing, how do we sort of figure out how these work all together to reduce the burden of malaria? So these are the questions that have been driving our modeling agenda and our methodology here. And the first thing I want to talk about is some specific results about what we learned about transmission that surprised us. This is about 6 years old now, but we've been studying this for a long time, trying to do evaluations for malaria vaccines, and we had these indications from the questionnaires that people were getting infected a lot from traveling off island. Because you can see that there's actually a ferry that goes from the island down to Rio Muni, which is not very far away, and a lot of people get on that ferry and travel back and forth. And we need to know what fraction of people are going back and forth. So we used the malaria questionnaire data in an investigation, and we actually made an estimate using that data of the fraction of malaria transmission that we thought was occurring as a result of travel. And this is sort of a method. So this is 3 ways of showing the same thing, but essentially our estimate was that almost all of the exposure in that downtown area is occurring as a result of travel off island. So the picture we're getting from this is that most of the malaria control we need to do on island should be focused around the periphery, what we call peri-urban areas. That's a term of trade, like it, we'll use it, or the rural areas around the outside. So this was a hypothesis, right? And how do we know if we're right? Well, it turns out the COVID epidemic came along and provided this really interesting opportunity. But remember, malaria is going up and down for other reasons. Rainfall, changes in rainfall, whatever. So a person who was, so I was on her thesis advisory committee, but she just came with, mad fat skills anyway. She came in and she did this difference of differences study. And I'm not going to do this justice. I hope at some point you'll get to hear Diana talk about her own work. But the main thing was that during COVID, it looks like malaria transmission went up everywhere. But it went up mainly in the areas on the outside. It didn't go up in the city, right? So we have this sort of increase in average transmission, but because of COVID, a reduction in malaria importation. So this was confirming for us that a great deal of the transmission that was occurring in the city center was probably due to malaria importation. And we still don't know how much. And we're still grappling with this question as we try to weigh sort of the effects of larval source management. This was sort of one of the ways we adapted, took advantage of a natural experiment, gathered information, and evaluated a hypothesis, right? So that we can come around and do a better job next time. The next thing is this idea of indoor residual spraying. I'm going to come and spray a bunch of houses. Remember, I talked about the relationship between coverage and effect sizes. Well, the budget doesn't cover universal house spraying. So every year they have to make a decision about how much, or about how much spraying to do, and then which houses to spray. And so they sort of initially started with various targeting schemes, and then at some point they wanted to know, should they be aiming for 80% coverage in as many places as they can, or should they not? And so they did a quasi experiment. It's an operational experiment. No difference ethically between what they would have been doing. They're just moving stuff around, so everybody's going to get some level of coverage. But now what they do is they assigned population units to either 50% or 80% coverage. That was their intent to treat. And then they went and evaluated all of this. So this is in review right now, and we're having problems with the bulletin of the World Health Organization because they want us to register this as a clinical trial. And we're like, look, it's an operational trial. If you're not going to accept this kind of research, then we're hosed, because we can't do randomized control trials, but we still need to learn about malaria. So what was the take home message from this is we couldn't see a difference in outcomes between 50% and 80% coverage. So now we're back to scratching our heads because we thought we should as to what's going to make a difference and how do we actually go about evaluating coverage targets, coverage strategies next year. And that's where we are right now. Okay, to answer some of these questions, and I'm done, we started delving pretty deeply into mosquito ecology and behavior. And so we started building models where we break up the life cycle now into compartments. So the behavioral state model, mosquitoes are blood feeding, resting, because it takes a while to digest the blood meal after they've taken it, sugar feeding, and they're doing this on point sets. And what we're discovering is that even with very simple rules for distributing resources, because we don't know what they look like, we can do analysis, but we wanted to know how to analyze these. First of all, it's very hard to build these models. It's very hard to analyze them. So we built software to do it so we can take a deep dive into mosquito ecology. And what we're discovering is some very interesting things. The take-home lesson here, it's a very simple one, is if an area is missing one of the resources mosquitoes need, they'll tend to leave it in search of that resource. And whether they come back or not is a matter of proximity to places where they fed on another resource. So you see that the co-distribution of resources on landscapes are what's driving mosquito behaviors and aggregation, and could be explaining hot pockets. A hot pocket would just be a place where mosquitoes have plenty of blood, sugar, and places to lay eggs so that they never leave, right? And you could have a very small area that has a very intensive transmission. So this is how we're trying to explain patterns like this and perhaps use them to guide control. I have gone probably enough, but I just wanted to say, if you guys have seen The Matrix, I want you to go back when he gets the advice to follow the white rabbit, and he goes to the door and he's holding a book. The title on the book is Simulacrum and Simulation. So what is simulacrum and simulation? It's a very interesting book, and this will take you to the Wikipedia page. It's a book on, it's kind of hard to say what it's about. It's about literary theory in some sense, but it's also about the way we use signs and symbols to communicate. So a simulacra is something that's a copy of something that never existed, right? So I've drawn a picture of a very realistic looking pig human in hyperrealism that never existed, and that's called simulacra. And his point is that what we, a simulation, of course, is what we do to try to mimic reality. And this point is actually that when we communicate in ordinary life, we often manage to never communicate any content because we're just passing along signs and symbols that end up being simulacra. They don't fill anything. It's very deep. You should read it. It's affected the way I think. And it's also affected the way I think about modeling, because if I'm going to go be honest with myself, if you look at those models that Ross McDonald developed, I don't know how to relate them to reality anymore. And if I wanted to relate them to reality, it's only as an approximating model to something that's very complex. If I'm going to do that, I need a formal system for making approximating models and evaluating approximating models. We don't have that. So we need to put metrics on distance metrics effectively on model spaces. So that's the next frontier. We're going to start doing that. And on the mosquito side, we think these graphs like this or approaches like this have taken us to a point where we can actually link up, very detailed behavioral data mosquitoes are doing, information about the landscapes to make a very good representation of what mosquitoes are doing and this concept of the sort of mosquito niche. So I'm now done. I hope there's time to answer questions. If not, find me, let's talk. So a couple of things that I'm really going to ask you for is you guys, a lot of you have experience doing adaptive management. You've seen me talk about this. You probably have opinions and you disagree about things. I would be delighted to hear how you disagree. I really need feedback, and I want to sort of be very interested in hearing what everybody thinks about some of these ideas. Am I rejecting parsimony? Did I just be, start as Tom Riddle and convert into Voldemort? Have I gone down into the dark arts too far? Or is this a really important idea that needs to get a more prominent viewing for student training. And just want to credit my team in Uganda, my collaborators in the Bioko Island Malaria Elimination Program who were here last week, and the funding that has made all of this possible. So thank you very much.
Thank you for the talk. We indeed have 6 minutes per question. And online folks, feel free to submit a question in the Q&A box.
I should put up a poll. Am I Voldemort? If I delved in too deeply into the dark arts, you won't even recognize me from the Tom Riddle who went to school with Simon and Ben.
Some questions, my parents, while we were, they had all these programs where, they basically went house to house looking for standing water. So it could be like an empty tire or gathering of grief or a bucket outside, and they would just basically have water, go search your house and surroundings, empty out standing water. They didn't need more than a pool for the mosquitoes to breathe and keep going. Is that something that's feasible again, but that's just such a great, these such great areas and with standing water everywhere?
It's actually what we're trying to do right now. Bioko Islands, they launched a larval source management program last year, and we have data from it that we're just now starting to analyze. The hard things though are what fraction of all productive sites are you actually treating? So you treat the ones that you see, but there can be some that are very difficult to find, and you need ways of sort of knowing where to look for things that you missed over and over again. So we have some ideas about how to do that, and the problem that we're coming up with is it's very difficult to catch adult mosquitoes in big enough numbers to evaluate pulses. So we need to sort of change the way we're doing trapping. And there's a long discussion to be had here, but you have to really give some serious thought to the costs and to the way that these metrics can change on you over time, because they end up being misleading. But the answer is this is exactly what a lot of countries want to do right now, but there isn't a lot of productive guidance about how to do it well or how to evaluate it. And so this is one of the areas that I think I need to fill in the gaps, because it's just something that's in my wheelhouse and it's not in a lot of other people's wheelhouses. Is to think about how to develop new metrics to measure, say, something like sensitivity. What fraction of productive habitats are you identifying? Or specificity. What fraction of habitats that you're treating do you not need to treat? Because, these are things inophalates would never just, or, those are the kinds of concepts that we need to, and then accuracy or effect sizes all need to be developed specifically for larval source management, probably in each place, kind of a bespoke approach. So, thanks for the question.
So speaking of advising governments, so how much do your models take into account the unfortunate fact there's probably going to be cuts to these programs that they didn't run more effective in spring? So you can have malaria as seasonal, every year. It's not necessarily be a reasonable understanding, right? Versus if you have a period of time of being really successful when funding dries out and you put it again. And the consequences of
Very good question. I mean, I could tell you lots of horror stories, but part of our goal is to condition people when we communicate about malaria control to warn them about this. So there's a lot of enthusiasm in Uganda now for malaria elimination. I don't know what they mean by it, and it's not what most people mean by it. But if they ever did eliminate, they have so many malarious neighbors, South Sudan in the north. And the sort of Turkana region doesn't have much malaria, but down around Lake Victoria on the east, and then DRC on the west, there's plenty of malaria coming across the borders. So if you did eliminate it, you still have to have vigilance in place to make sure that you locked it up. We've written a lot about this under the sort of malaria elimination, long-term planning, strategic planning, and evaluation of programs that worked when they shouldn't have. So it turns out that if you do actually ever eliminate malaria, it tends to not come back. But elimination in this case means that you haven't seen 3 cases epidemiologically linked. By the way, we had long discussions about definitions of elimination. And but essentially you mean that you've eliminated all local transmission. And that you can prove to the WHO that your surveillance systems are good enough that if there was transmission going on, you'd know it. If you ever get that far and eliminate malaria, it tends to not come back. If you get partway there, it tends to come back with a vengeance. So there's a question of what happens in that transition period from almost eliminated to eliminated that really makes a difference for, we call it stickiness. And I've got a few papers about it that I could sort of look at the historical data. It's a fascinating thing that we don't understand very well. And there could be evolutionary hypotheses, like it could be that the parasites have adapted to the local mosquito populations. So that's one that has been examined. There might be some evidence for that. Could be there are other things that are more ecological going on, but it could be economic. Everything we saw was due to sort of the removal of malaria caused a lot of economic development, and rich people don't get malaria. They just don't. It's not that expensive to keep yourself continuously chemoprotected against malaria. So if you want to never get malaria, you can never get malaria, but you have to have enough money to do it and knowledge about it. So knowledge and money are enough to keep malaria out of a place. Maybe that's what's in. I mean, it's just, or the places that sustain malaria tend to be rural areas, and most of the people that come visiting from far away come to urban areas. Think about all of the things that would sort of make malaria elimination stable, maybe shouldn't work in a well-mixed model, but actually work in reality. And we tried to enumerate these in this paper and did some analysis. I mean, all of these things we're looking at are very complex things, and so we're doing inference for complex things, and it's always highly soft in some sense, right? But we're trying to develop guidance for future malaria. So that's where we ended up.
It's just sort of like the place where they did eliminate the basic spring DDT.
Yep, it worked. It worked really well.
I was at a game park in Bolivia, spraying on that house everything about chatting to. And he basically said, "Yeah, I'm spraying this stuff, but this is from a TDT. Don't bring it back to DDT stuff." I'm like, so that's like 40 years off we back.
Yeah, there's a long story. So Actelic actually seems to work as well, but it's more expensive. DDT, once we did the ban in went in the register, I think 1971, and the US stopped producing it, and then that kind of went, the European countries kind of banned it as well. There was a long history of this. Kept going. It was produced in India and Mexico for a long time and used in various things. And then in this most recent era, like Uganda wanted to use DDT for indoor residual spraying. Europe threatened to boycott their fruit. So the sort of political will, but actually, when you look at DDT as an environmental contaminant, and this discussion happened in the late 1960s and early 70s, it's a different thing than if you're spraying it on the houses, where there's not much environmental contamination. So there was the people who were involved in the global malaria eradication program made a plea to carve out a public health exemption for DDT. And they were not listened to. And so, I mean, this is where politics and advocacy and everything come together in some of these really interesting stories where maybe that wasn't the best thing, is to not carve out a public health exemption. The argument was if we carve out a public health exemption, which is probably right, people are going to find their way around it and keep using it now. And so that was some of the debates that happened then and have gone on and on since then.
I'm ironic when we went to US, Africa, and then that was also DDT. And that's the thing. But it's
Yeah, the history of each of you have to look at each country has its own history. So why is the CDC in the United States in Atlanta? It was founded to plan for resurgent malaria after the troops came home from fighting in malarious countries at the end of World War II. Did we get any resurgence? No, not much. And so the malaria was on its last leg and was in the years after that was sort of completely eliminated. But the history of how we got there is really a US specific set of things, right? Because there was malaria control during the CCC, during the, the response to the Great Depression. We give everyone a shovel and $1 a day to go do something. And so there was malaria control projects done through the CCC, and there were other things happening because sharecropping effectively ended as an economic institution, as those jobs in manufacturing, make things for the war, hold people out of rural areas and into the cities. So sharecropping ends overnight. So that, you look at it, looking at big changes in malaria, they do happen. But there's sometimes mosquitoes are evolving, sometimes the mosquito human interface is evolving, sometimes the economic situation is changing, there are big demographic changes or economic changes. And those are, in Africa, probably we've underappreciated the role of improvements in housing quality over time. As people have just sort of steadily improved their houses, making it harder for mosquitoes to enter. So these are things that we really need to take into account when we do our analysis because they matter. And they can you can misinterpret your data if you don't have them in there the right way. They're fantastically interesting questions to be addressing.
Yeah, I have to stop here.
Okay.
It's like five
Thank you
minutes
for
past.
listening.
Thank you everyone for coming and we hope to see you next. And if you have further question, please reach out.
There's in Zambia, huh?
I don't take time, but there's for our.